how we can change it to achieve immunosuppression. So we have seen earlier various agents like tacrolimus, sirolimus, and cyclosporine. Now let us go for the next type of drug. These are the inhibitors of cytokine gene expression. Examples of these are corticosteroids. Examples of these corticosteroids are again many: prednisone, prednisolone, methylprednisolone, dexamethasone, cortisol, hydroxycortisol. There are many such agents are there, and these are having variety of action. We have taken very few examples, but if you remember last year we have seen budesonide, etc. in respiratory system. So there are many such corticosteroids. They have both anti-inflammatory action and immunosuppressant action. Rather, immunosuppressant action is helping in its anti-inflammatory action. The mechanism is simple. These agents binds to glucocorticoid receptors. These are the nuclear receptors, and the complex interact with the DNA to inhibit the gene transcription, particularly for inflammatory genes or the gene which is resulting into processing of inflammatory mediators. Decrease in production of these inflammatory mediators like prostaglandins, leukotrienes, histamine, then platelet activating factors, bradykinin. These all are reduced because of reduction in this production like interleukin 1, interleukin 2, interferon, TNF alpha. So all of reduce their synthesis production decrease. We have seen their role earlier. They act as a chemotaxicant. We have seen chemotaxis. They play important role in inflammation mediation. And we have seen interleukin 1, 2, they are playing important role in the proliferation and differentiation of various T cells. So, all in all, because of this variety of actions, they are playing important role in inflammation as well as our control of inflammation, and they are playing important role in immunosuppression. So this is in addition to that, they will stabilize the lysosomal membranes. So lysosomal activity or hydrolyzing activity of lysosomes should not be there. That is, or what we can say, cell toxicant activity will not be there. They will also decrease the generation of various immunoglobulins like IgG, nitric oxide, and histamine. Then they will inhibit the antigen processing by the macrophages. Thus, they will reduce the engulfment. They will suppress the T cell helper function. They will decrease T lymphocyte proliferation. We have seen actually basic mechanism behind that was from the point of view of interleukin 2 synthesis. So these are a variety of action, and it is doing the job manifold, and therefore they are very effective. They are very useful because they can be administered by variety of routes, maybe orally or parenterally. They causes the suppression of response to infection. That is, we have seen that infection is there, then the cells are there. That is CD4, CD8, or various T cells are there. They will take, if antibodies are found, then antigen antibody reactions will be there, and this will cause initiation to response to infection by either cellular immunity, that is engulfment of microorganism, or producing antibodies against it. So overall, all these kind of responses to infection will be suppressed. Then additionally, anti-inflammatory action is there. Immunosuppression we already seen, and since it is a hormonal type of agents, it plays various metabolic effects also, and most of these adverse effects are having a long-lasting effect. Therefore, it has to be used very carefully. Uh, they are first-line therapy for solid organ allografts and hematopoietic stem cell transplantation. They are also useful in autoimmune diseases like refractory rheumatoid arthritis, systemic lupus erythematosus, and asthma. Then they are useful in acute or chronic rejection of solid organ allografts. The problems are adrenal suppression, or what we call it as a HPA axis, that is the hypothalamus pituitary and adrenal cortex, that is we call it as a HPA axis. So that HPA axis is disturbed, there is a very good feedback control which is existing. That whole is changed, and because of that it causes adrenal suppression. It causes osteoporosis, then hypercholesteremia because of its metabolic effects on variety of places, and because of that it causes this impact. Then they are responsible for hypertension in many. It causes blurring of lenses that result into cataract. It can give rise to infection of sort of super infections, that is because of a reduction in the immunity. Even the opportunistic microorganism which otherwise were not pathogenic can become pathogenic now because of that. Then gastric ulcers it can cause, because again its impact on the prostaglandin and other synthesis. It has got CNS effect. It can affect the growth failure. It can delay the puberty. So these are all variety of effects as adverse effect it is showing. Very very important thing about this agent or these corticosteroids is they do have comparatively, still despite of all these adverse effects, they are comparatively safer than sirolimus, tacrolimus, or other agents, because still they are better tolerable or lesser adverse effects comparatively than this. One more important thing is there, whenever these drugs are administered, these drugs, what's your doses are administered, but at the end, when they are to be withdrawn, they should be gradually withdrawn, or we call it as a dose tapering. So the dose has to be tapered slowly, it cannot be abruptly withdrawn. Just like in antibiotics, somebody or painkillers, when there is no pain, somebody can stop painkiller abruptly. So in these cases, the drugs cannot be stopped abruptly. So this is a very very important thing. This drug always has to be tapered slowly. If earlier, suppose 40 milligram per day was the dose, then slowly it has to be reduced to 30 or 20, and gradually it has to be stopped. So at every stage, there shall be stabilization, and then slowly it should be reduced. This is very very important, particularly its problem with the adrenal suppression. That is, or HPA, as I told you, that is hypophyseal or and hypothalamus pituitary adrenal axis. So this axis is very very, HPA is very very important in feedback regulation, and therefore when the external drug is given, then this feedback is disturbed because much more amount of drug is present. So all of a sudden, if it is there, then rebound increase can be there because of this feedback mechanism. So it is very important. Next type of drugs are cytotoxic drugs. Example of these are inhibitors of purine or pyrimidine synthesis. They are also, we call them antimetabolites. I think we had a good discussion on antimetabolites earlier. Example of these are azathioprine, then myclophenolate mofetil, deflunomide, methotrexate, and other agents like alkylating agents. These all are cytotoxic drugs. As its name indicates, cyto and toxic. Cyto means cell, toxic means toxic. The drugs which are toxic to the cell, those which can kill the cell, these are the cytotoxic drugs. So, as the name indicates, they do have comparatively higher adverse effects. What they kill, which cell they kill the cells which are actively taking part in immunological reaction, and because of that, they are killing them. Because of killing of these cells, they will not able to perform the immunological mechanism or any kind of immunological activity, and because of this, these drugs will act as a side and what we can say immunosuppressant agents. So first in that class is azathioprine. Most of these drugs you will find that they are either antiviral or anti-cancer drugs, because antiviral drugs are also very, very close to that of normal physiological biochemicals, so that the viruses do not receive. So these are the derivatives. Azathioprine is a derivative of mercaptopurine. It is a pro. It is cleaved to a 6 mercaptopurine, then to 6 mercaptopurine nucleotide is there. Then thioinosinic acid is there. Uh, just like purines are metabolized in the body, that is, purines are synthesized, then purines are glycosylated or glucose is attached, phosphate is attached. So from that, suppose if it is adenosine or guanosine, these are becoming adenosine monophosphate or triphosphate, like that. So similarly, here sugar moieties will be attached, nucleotides they will become nucleotides, and then they will get incorporated into the normal purine metabolism. So they will inhibit the de novo synthesis of purines, which is required for lymphocyte proliferation. So similarly, instead of normal purine, this mercaptopurine will be getting inserted in the nucleic acid at the place of adenosine or guanosine, that is adenine or guanine. So thus they will be there. It will produce a faulty product in gene activity, and therefore it will not able to do the job which is required. It will prevent the clonal expansion of both B and T type of lymphocytes. So this is how the mechanism is there. As I told you, this was a base, mercaptopurine base. Then the hypoxanthine guanine phosphoribosyltransferase transferase will cause the attachment of sugar molecule and phosphate molecule, and this will produce thioinosinic acid, that is thio-IMP. This IMP will cause many activity. It will inhibit the feedback inhibition of phosphoribosylglycine synthesis. Then this inosine monophosphate will give rise to uh, AMP and XMP, that is xanthine monophosphate or adenosine monophosphate. So this will be inhibition, thio-GMP will be produced, and that will be incorporated in DNA. That will not be a correct one, and then that will result into termination of protein synthesis and such important metabolic process, which are important for the T cell and B cell or lymphocytes. So this is how this will be acting. It is orally or intravenously can be administered. It is widely distributed, but it cannot cross blood-brain barrier. It is metabolized in the liver to 6-mercaptopurine or to thiuric acid, which is inactive by xanthine oxidase enzyme. It is excreted primarily in urine. Co-administration of allopurinol with diazothioprine may lead to toxicity, as xanthine oxidase will be uh, inhibited by allopurinol. If you remember gout, allopurinol is the drug which is used in the treatment of gout, and that causes inhibition of xanthine oxidase, where more xanthine oxidase is resulting into more purine metabolism and deposition of uric acid. So this is how these agents, allopurinol is there, and allopurinol is there, then it can cause toxicity. So what are the uses? It is used in acute glomerulonephritis, systemic lupus erythematosus, or rheumatoid arthritis, and Crohn's disease. Adverse effects are mainly bone marrow depression. It can cause lipopenia, thrombocytopenia, gastrointestinal toxicity, hepatotoxicity, and increased risk of infection, because all immunosuppressants, they will have a very typical peculiar adverse effect will be most of this type of activity, that is decreasing the cell count, is one thing, GI toxicity is another thing, and third is increased risk of infection. These three will be very common in most of the agents. Then next is mycophenolate mofetil. It is a semi-synthetic derivative of mycophenolic acid from a fungus source. The prodrug is hydrolyzed to mycophenolic acid. It also inhibits the neosynthesis of purines. It is a potent inhibitor of inosine monophosphate dehydrogenase enzyme, that is IMP dehydrogenase, that is crucial for purine synthesis. It will deprive the proliferation of T cell and B cell cells of nucleic acid. That is, nucleic acid will not be properly formed, and thus, even if the cell are differentiating, it will be without this proper nucleic acid, and therefore will not able to function properly. So this is how this agent acts. That is inosine monophosphate. This mycophenolate will inhibit, or it will compete for the same mechanism, and instead of that, this mycophenolate will cause xanthosine monophosphatase, which will result into a improper guanosine monophosphate. So it will blocking the formation of guanosine monophosphate, and it deprives rapidly the proliferating T and B cells of a key precursor which is required for nucleic acid synthesis. So if this nucleic acid synthesis is not done properly, or it is not there, then the differentiation will not be there, or the cell division will not be there. So ultimately, it will cause reduction in the T cell B cell count, and if they are less, then definitely we know about that it will decrease the immune reaction, or its participation in immunology. So it can be given orally, intravenously, or intramuscularly. It is rapidly and completely absorbed after oral administration. It undergoes first pass metabolism to give the active moiety, that is mycophenolic acid. It is extensively bound to plasma protein, metabolized in the liver by glucuronidation digestion. It is excreted in urine as glucuronic conjugate. The dose is 2 to 3 gram per day. So it is again used in solid organ transplant for refractory rejection, steroid refractory hematopoietic stem cell. I hope you understand this word. Steroid refractory means where the glucocorticoid cannot work. We have seen glucocorticoid as there, but where it is not. That also suggests one important thing is glucocorticoid are having first preference over this. But if it is the refractory to steroid, okay. So in again solid organ transplant for refractory rejection, that means that if it is refractory to the first line treatment, then these are the second line treatment which are available. It can be combined with the prednisone as an alternative to cyclosporine or tacrolimus. Then rheumatoid arthritis and dermatological reaction, in that also it can be used. But again, I repeat that as we are going ahead, most of these are more toxic and having more ADRs. So most, though glucocorticoid has got more adverse effects, still in all these group it is safest one. So when it is not appropriate, or when it cannot be used because of one or other reason, because of some specific adverse effects, or maybe because of the refractoriness to the treatment, then one can go for other drug or combinations. Uh, ADI, same as we have seen GIT, toxicity, nausea, vomiting, diarrhea, abdominal pain, leukopenia, neutropenia, lymphoma, it can cause. It is contraindicated during pregnancy, and definitely one more is always there, chances of infection. Then leflunomide is a prodrug, active metabolite. It undergoes enterohepatic circulation. It has got long duration of action, can be given orally. It has got anti-metabolite immunosuppressant action, pyrimidine synthesis inhibitor, approved only for rheumatoid arthritis and not in other cases. Uh, Adverse effect, it causes elevation of liver enzymes. Elevation of liver enzymes indicates the injury or problem to the liver. Okay, these are, I hope you might have studied them in pathophysiology, various enzymes like serum ALT, AST, and alkaline phosphatases, etc. So these are various important liver enzymes. If they are elevated, that means there is a injury or damage to liver. They can also cause renal impairment, can cause teratogenicity used in pregnancy. You know about teratogenicity now. What Pocomelia, which you searched earlier with thalidomide, it is one of the teratogenicity. Okay, that is malformation of body is a teratogenicity. Then cardiovascular effects like tachycardia. So because of so many adverse effects, it is not very widely suggested, recommended, or prescribed. Only in case of rheumatoid arthritis can be there. Then it is a folic acid antagonist. That is, it blocks the conversion from folic acid to tetrahydrofolate. Then it is orally, parenterally can be given. It is excreted in urine. Already I told inhibit that hydrofolate reductase required for folic acid activation or conversion to tetrahydrofolate acid. Because of that, which tetrahydrofolate acid is very very important coenzyme for the one carbon ion carbon atom transfer, which is important in requirement during various replication as well as protein synthesis activities, and therefore it will inhibit the replication. That means DNA to DNA will not be there, then it will inhibit transcription and translation, that is RNA and protein synthesis will be inhibited. It will interfere with the T cell replication. So it can be used in rheumatoid arthritis, psoriasis, and Crohn's disease. Uh, then also in graft versus host disease, it can be used. ADRs are nausea, vomiting, diarrhea, alopecia, bone marrow depression, pulmonary fibrosis, and renal and hepatic disorders it can cause. So these are the important ADRs these methotrexate is having. So how these drugs are acting, I hope you have seen it earlier. So from anyway, either from diet or intestinal flora. What is intestinal flora? That we have got microorganism in intestine. These are the beneficial microorganism, or they are the microorganism which are helping human body. So they are doing many synthesis, many vitamin synthesis. One of the important role they are playing is the folic acid synthesis. So this folic acid is to be converted to tetrahydrofolate, and thus this tetrahydrofolate, uh, that is the reduced form, that will be important in the synthesis of adenine, guanine, thymidine, methionine, serine. So these are various important amino acid proteins or nucleotides. So these are important. So in case of toxicity, we have got antidote, that is leucovorin, that is it is N5 formyl tetrahydrofolate, or leucovorin or folinic acid. It is called as, it is converted to the methylene tetrahydrofolate, and therefore bypasses the inhibition reductase. So in emergencies, this agent can be used, particularly when the toxicity is very high and it has to be reverted back. Then this leucovorin can be used. That is called as a leucovorin rescue, because it doesn't require enzyme for activation. So enzyme, the drug has blocked the enzyme. So it let the enzyme block. It will be directly providing the final metabolite which was required. Next kind of drug is cyclophosphamide. It is a chelating agent. When we shall see anti-cancer, at that time we shall see the mechanism in more detail. This is a prodrug. It is activated into phosphamide. It is given orally and intravenously. Destroy proliferating lymphoid cells. Then anti-cancer and immunosuppressant action it is showing. It is effective in autoimmune diseases like rheumatoid arthritis, systemic lupus erythematosus, and autoimmune hemolytic anemia. So these are various uses the drug is having. There are many adverse effects: alopecia, hemorrhagic cystitis. These are the from various the hemorrhages will be there. Bone marrow separations, GIT disorders like nausea, vomiting, diarrhea, even ulcers it can cause. It can cause sterility, that is a testicular atrophy and amenorrhea it can cause, and therefore the man or woman may suffer from sterility. That means uh, they will not able to have the what we can say fertilization, and thus it will cause the problem in the birth in their family. So this will be the problem. That is cardiotoxicity again is one of the toxicity it is causing. The next kind of drugs which are very recent one, actually in last 10-15 years they have grown, maximum up to 20 years take. So the growth is there, and very very popular. They are comparatively very rapidly being used. So what are these agent doing? They can block the T cell surface molecules which are involved in the signaling immunoglobulins. So because of this signaling, there is a proliferation, there is a recruitment or chemotaxis, and all these activities are there. So if this cell signaling is blocked, then it can block all the further cascade of reaction. So what are the types of these agents? These are anti-lymphocyte globulins. That means the globulins which are globulins are the as such blood proteins, and these blood proteins are albumin globulin, if you remember. And out of that, these globulins they are playing very very important role in antibodies. Even all natural antibodies are there of globulins. So if you remember IgE, immunoglobulin E, IgE, IgM, IgD, there are a variety of such globulins are there. On similar platform, on similar basis, they are externally formed by biotechnological measures, and they are administered into body. So these are a variety of agents in that anti-lymphocyte globulin, then anti-thymocyte globulin, then Rho D globulin, Rho D immunoglobulin. These are against the problem, which is because of the RS factor related problems which is there in fetus, in fetus. That is that there are many fetus they die because of the blood grouping are the problem. You might have studied the disease that is erythroblastosis fetalis. Okay, that is erythroblastosis fetalis of the newborn, very common disease because of the problem in the mother's and fetus blood group, particularly RS factor. Then other in that is basilicimab, then daclizumab, then infliximab. See, this mab always means monoclonal antibodies at the end. So the name itself indicates these are the monoclonal antibodies. How these are prepared by, actually this was old methods. Initially they are done with the horses, rabbits, with human lymphoid cells producing mixture of polyclonal antibodies. They are directed against a number of lymphocyte antigens, and from that, then once it is there, then it can be for mass production. It can go for biotechnological production, that is insertion of these genes to some other smaller species or microorganisms, and then this microorganism will be synthesizing this kind of antibodies. This whole mechanism is called as a biotechnological process. So many drugs which were earlier from animals origin nowadays are being produced biotechnologically. Example, when we were studying about insulin oxine or porcine, like that. That is from pig or bull. So oxine like that. Nowadays everything is available. And they were different. They were having adverse effect. Nowadays, which is available, it is biotechnological human insulin. So it is similar to that of human, and it is being produced by microorganism. Because of the genes which are inserted, or their genes are modified in the microorganism, and from that nowadays such are there. So this can be done with biotechnology, and antibodies can be prepared accordingly. Then other technology is hybridoma technology. It produces antigen-specific monoclonal antibodies from homogeneous specific. They are producing by fusing mouse antibody producing cell. with immortal malignant plasma cells. Hybrid cells are selected, clone and selectivity of the clone can be determined. Then already I told about this. This is called as a recombinant DNA technology that is used to replace part of the mouse gene sequence with human genetic. And the antibodies from mouse they contains muro in name. Example, they are called as a muromonab. That is muro monoclonal antibodies. So like that, they are the humanized antibodies contain ZU or XI in their name because they are humanized. So like that, how the nomenclature of these antibodies is are there. So what are various types of antibodies? So polyclonal antibodies, poly means multiple. So polyclonal antibodies are obtained from plasma or serum of the horse hyperimmunized with the human lymphocytes. They binds to the surface of circulating T lymphocytes, which are phagocytocytes in the liver and spleen. They are giving the lymphopenia and impaired T cell responses to the cellular immunity. Then what is kinetics? They have to be given by intramuscular or slowly infused intravenously. If it is rapidly there, then they themselves can cause very severe hypersensitivity reaction. Half-life extend from 3 to 9 days. They are combined with cyclosporine for bone marrow transplantation. They can use for treatment of acute allograft rejection and even steroid resistant rejection. They are useful. Adverse effect already I told you antigenicity, therefore slow injection, IV injection, and it is always better to be tested before administration. If there is no antigenicity, then with the time it will cause lipopenia, thrombocytopenia, it will cause risk of viral infection, and anaphylactic and serum reaction that is fever, chill, flu-like symptoms. Out of that, anaphylactic is always very dangerous one. Again, during penicillin also we shall see it because it is very sudden, rapid, and person can go in shock and many times cannot be saved. So these are the major problem these agents are having. So indeed, first is muromonab CD3. So this is murine. Murine means from mouse. It is there. Monoclonal antibody. It is prepared by hybridoma technology. It is directed against glycoprotein CD3 antigen of the human T cells. Then it has to be given intravenously. It is metabolized and excreted in the bile. How it acts? It binds to the CD3 proteins on the T lymphocytes. This is called as the antigen recognition site, and on that if this binds, then it will not able to perform the job as antigen recognition site. So it will cause leading to transient activation and cytokine release followed by disruption of T lymphocyte function, the depletion of T cells, and decreased immune reaction. Prednisolone diphenhydramine are given to reduce the cytokine release syndrome simultaneously, which is occurring because of this. Because initially it is increasing the cytokine release and then there is a disruption. So too much cytokine release can be again a problematic one, and therefore these agents are given to reduce the cytokine release syndrome. What are the uses of muromonab? That is, it is used for the treatment of acute renal allograft rejection and steroid resistant acute allograft. It is used to deplete the T cells from the bone marrow donor prior to transplantation. Adverse effects, almost all are common if you have seen earlier. Anaphylactic shock, fever, CNS effect like seizures, infection, cytokine release syndrome that causes flu-like illness to shock-like reaction. Shock many times because of very severe vasodilation, so we call it as a hypotensive shock. So severe hypotensive shock it can cause, and it can very lethal also. Then other type of immunoglobulin is RO, that is Rho or D immunoglobulin. We know about, I told you about this. It is a concentration solution about human IgG1 containing higher titer of antibodies against Rho, that is RhD antigen for red cells. So when they are given to Rh negative mother within 24 to 70 hours of the delivery of Rh positive baby. This will this will prevent the hemolytic disease of the next Rh positive babies. So this is called as erythroblastosis fetalis. In Totora, the details are given. Those who are interested can go to it. The adverse effects are local pain and fever it causes. Then next type of these are the basiliximab and daclizumab. So as the name indicates, XI MAB and CDU MAB, these are the humanized one. So they are obtained by replacing murine amino acid sequences with the human one. So this XI and ZI, they are giving ideas, that is chimeric human mouse. Chimeric means these are humanized one. Uh, so these are the chimeric human mouse IgG, 25% murine and 75% human protein. While in daclizumab, it is mostly humanized one. So 90% of human protein is there. If more is humanized, then less antigen chances are there. Same thing if you remember you have seen with the insulin. So oxine, porcine, insulin were having more hypersensitivity reaction than that of the human one, because human is more acceptable. So it has got less antigenicity and longer half-life than the murine antibodies. They are acting on interleukin 2 receptor. They are acting as an interleukin 2 receptor antagonist. Both are having action anti-CD25. They bind to the CD25, that is alpha subunit chain of interleukin 2 receptor on the activated lymphocytes. They will block interleukin 2 stimulated T cell replication and T cell response system. The basiliximab is more potent than daclizumab. They are given intravenously. The half-life of basiliximab is 7 days. Daclizumab is around 20 days. Well tolerated, only GIT disorders. Given with the cyclosporin A and corticosteroids for prophylaxis of acute rejection in renal transplantation. Only thing is these are very costly agents because these are new also. These are under patent and very big research expenditure has been made on this, so they are comparatively very costly. And if somebody compares with the glucocortical, they are exorbitantly costly. Then other monoclonal antibodies are infliximab. It is also chimeric human mouse immunoglobulin G. It is directed against TNF alpha. It is approved for ulcerative colitis, Crohn's disease, and rheumatoid arthritis. Then next is omalizumab. It is again humanized monoclonal IgG against IgG. It is directed against the Fc receptor on mast cells and basophils. It is approved for asthma and steroid refractory patients. Then interferons, there are three families are there. Type 1 interferons, interferon alpha and interferon beta. These are acid stable proteins. They act on the same target cell receptor. And their synthesis is induced by the viral infection. Whenever there is a viral infection, body produces more and more interferons. The leukocyte produces interferon alpha, and the fibroblast and endothelial cells, they will produce interferon beta. Then other interferon, that is type 2 interferon, it is interferon gamma. It is acid labile. It acts on separate target cell receptors. It is produced by activated T cell lymphocytes. Then next in that is immune enhancing. It increases the antigen presentation with the macrophages, natural killer cells, cytotoxic T lymphocytes activation. Interferon alpha and beta, they are effective in inhibiting cellular proliferation. So alpha and beta are more effective than interferon gamma in this regard. So they are useful in the treatment of certain infection like hepatitis C, in autoimmune diseases like rheumatoid arthritis, certain form of cancer like melanomas, renal cell carcinoma. They are useful in the multiple sclerosis, particularly interferon beta is useful in that. in the reduction in the rate of exacerbation of multiple sclerosis. Then fewer chills myelosuppressions are some of the actions. Then next is thalidomide. I hope you people have gone through the thalidomide after our discussion earlier, phocomelia and all. Actually, thalidomide was approved as a sedative drug long back in 1930s, and it was believed to be very good sedative agents, but it was not properly tested during toxicological studies. And then it was found that the females, particularly in Japan, it was very prominently found that wherever females has consumed it during pregnancy, they led to the delivery of children having phocomelia. So it was classified as teratogenic. There are lot many things went on. Many inquiries were there. The drug was banned and it was not there. Later, in few again last some years, it was found that it is a immunomodulatory action. Now here immunomodulatory means at some places it causes immunosuppressant, at some time it causes immunostimulation. So these are such agents which are actually immunomodulatory, that sometimes they causes immunostimulation at some places, and at some places it causes immunosuppression. So they are inhibiting the TNF alpha, then it reduces the phagocytosis by neutrophil. It causes increase in the interleukin 10 production. They are useful in the myelomas, in rheumatoid arthritis, in graft versus host diseases, in leprosy reactions, in treatment of skin manifestation of the lupus erythematosus. So these are places where it is required as a immunosuppressant. So now if we go for a brief review about various drugs, then from the beginning we can understand where the drugs are there, how it is available. So what are the steps in various immunological process that either microorganism which is presenting itself, or any other such antigen which is there as a, or which is presenting itself as antigen, will give rise to activation, that is it will act on the T cell receptor. So there are drugs which will bind the which will inhibit this. These are the antibodies like miromonab CD3. It will block this, and because of this anti-thymocyte globulins or miromonab, there will be destruction of T lymphocyte. Destruction of T lymphocytes will cause the uh, reduction in this immune reactions. Simultaneously, it can cause ADR. It will cause profound immunosuppression, and it can cause cytokine release syndrome. Then next step in that is T cell receptor. Once the antigen binds to T cell receptor, there is activation, and it causes the steps, variety of steps, that is activated calcineurin or activated this nuclear factor activation, then interleukin 2 gene proliferation. This all we have seen, and we have seen various drugs like cyclosporin, tacrolimus, etc. They are acting. They will block the calcineurin and inhibit the interleukin 2 synthesis, block this process. The adverse effects are nephrotoxicity, neurotoxicity, hepatotoxicity. These are various. The other one, tacrolimus, can also cause diabetes. So these are drugs are there. Then interleukin 2 is released uh, as a part of immune reaction. So we have got drug which is antibodies again, that is basiliximab and daclizumab. They will block the interleukin 2 receptor, and the adverse effect are GI disorders. Now, if interleukin 2 receptor is bound by this interleukin 2, then it will cause the action. So we have got sirolimus, which will block it, which will block the cytokine stimulated cell proliferation. We have seen it as it acts on mTOR, mammalian target for rapamycin. On that, sirolimus will be acting, and it will The adverse effect of these will be hyperlipidemia, thrombocytopenia, leukopenia, etc. Then, if it is not done, then interleukin 2 will cause progression of into cell cycle, and this progression into cell cycle will result into cell proliferation, and this cell will participate in the human reaction. This is what the mechanism is. So azathioprine, mycophenolate, morpholate will act on this purine synthesis. They will inhibit it, and thus will not allow cell proliferation. So this is what a glimpse of or the picture of how the drugs are playing important role at various stages. More than one step if we block, we can get the synergic action. So where we are using, see, these are various autoimmune disorders and what are the preferred agents. Example is acute glomerulonephritis. So we can use prednisolone, mercaptopurine, cyclophosphamide. Then autoimmune hemolytic anemia. So in that, the drug which are preferred are prednisone, cyclophosphamide, mercaptopurine, azathioprine, high dose deep globulin. For transplant, cyclosporine, azathioprine, prednisolone, ALG, tacrolimus, that is anti-lymphocyte globulins. For liver, cyclosporine, prednisolone, azathioprine, tacrolimus. For bone marrow transplant, cyclosporine, cyclophosphamide, prednisolone, methotrexate. Then anti-lymphocyte globulin, total body radiation can be done. Thymocytes are the cells that develop into the thymus and they serve as a T cell precursor. So we have seen various agents as anti-thymocytes, and many times that happens. What this thymocyte means. So these are all antibiotics. How they are acting. Polyclonal means having more than one binding domain, so that it can bind with more than one type of antibody. While monoclonal are the antibodies which will able to bind very specific and single type of antibody or antigen. Sorry. So this is how there is a difference in polyclonal multi. So we have seen this variety of antibodies, anti-CD3 like merovinov and daciclizumab. One more there that is in that is Campact. One is that is altezumab is there. Then we have seen infliximab. The other one are etanercept, adalimumab. These are a variety of monoclonal antibodies which are there. Then LFA1, that is lymphocyte function associated activity inhibitor is there, that is ifalizumab. So these are a variety of antibodies nowadays coming, and maybe in future you will find more and more such antibodies which are there because they are very specifically targeted, and therefore comparatively they are having lesser adverse effect. Only biggest problem with these are they are very costly, and they can be afforded only by those who are either having insurance or can afford them. Then ATG, we have seen antithymocyte globulin. These are purified gamma globulin from serum of the rabbits, which are immunized with thymocytes, cytotoxic to lymphocytes and block the lymphocyte functions. And they are in the immunosuppression transplantation. They are treatment in the acute transplant rejection and hypersensitivity, risk of infection and malignancy. Then CD3 we have seen just now. That is, it blocks the antigen recognition and cell signal and proliferation, and thus it reduces the immune reaction mechanism. We have already seen this is acute organ transplant rejection. Toxicity is cytokine release syndrome, high fever, chills, headache, tremor, myalgia, arthralgia, weakness. These are the cytokine release syndrome is there. We have seen that simultaneously we can combine prednisolone so that we can reduce the cytokine release syndrome. Okay, this is a very common complication which is there. Use of anti-T cell antibodies like ATG or OKT3. The pathogenesis is that antibodies bind to T cell receptor. They activate the T cells before they are destroyed, and because of that, the cytokines are released. by these activated T cells, and they will produce the systemic inflammatory response similar to that of found in severe infection characterized by hypertension. Those who have read details about COVID nowadays, they might have, or the various drugs which are used in that, some immunosuppressants are also used. This is sort of homework, you study about it. And it has been found that they are used for the cytokine storm. So those who want can just search on what is cytokine storm in COVID and what are the drugs which are used for the treatment of this cytokine storm, which agents are being used. Okay, this you can just as an assignment, immediate assignment. Okay? Then these are the, already we have seen daclizumab and bevacizumab, how they are acting, and then various anti-TNF agents like infliximab, etanercept, and adalimumab. These are other agents which are there. So how these are acting, how these are working. So these are the monoclonal. This is the normal one. That is, they are binding to the cytokine receptor and then they are giving inflammatory signal. While in other cases, these monoclonal antibodies will bind with them and they will not give the drug, give this cytokines available for the cytokine receptor, and thus they will not able to bind with the cytokine receptor. So this is how these agents are acting on that. Okay. Uh, the other drugs and some of the immunosuppressant agents we shall take, sorry, immunostimulants we shall take